This podcast is brought to you by the Albany Public Library main branch and the generosity of listeners like you.
What is a podcast?
God, Daddy, these people talk as much as you do. Razib Khan's Unsupervised Learning.
Thanks for listening to the ungated version of the Unsupervised Learning podcast. If you want to read some essays on some of these topics, please check out razib.substack.com. Again, that's razib.substack.com. Thank you.
Hey everybody, this is Razib Khan with the Unsupervised Learning podcast, and I'm here with two returning guests, multiple returning guests that are well known to you guys, John Hawks and Chris Stringer. Paleoanthropologists, bones guys who know quite a bit of genetics or have been involved, in and around the genetics field as well. So, they've come on multiple times. I've introduced them. I think you guys know who they are. So in the interest of time, I'm just going to jump into what we want to talk about. So mostly I want to talk about Denisovans, Eastern Eurasian hominins. I'll use that generic category because things are still a little unsettled, although they're getting more settled. So we have a paper, Chris, that you were on, the phylogenetic position of the Jungian cranium elucidates the origin of Homo longi and the Denisovans. Can you just talk about, since you're on the paper, I would like you to talk about what your general interpretation of the paper is, what it means for us, and then John, why don't you respond? Because I know you wrote a post about it that was, I thought, pretty interesting. So.
Okay, yeah. So the Jungian 2 fossil found 1990, so it's been around a long time, but obviously distorted, crushed in the ground and distorted, which has hampered interpreting it. Generally, it's been called Homo erectus, some kind of variant of Homo erectus, sometimes archaic Homo sapiens, and it's about 1 million years old. So the site is pretty well dated by paleomagnetism and also the fauna there, which is quite a rich fauna. So it's pretty well dated to around 1 million. So it's quite important. And as I say, the default view would be that this probably would be Homo erectus, that kind of date. Anyway, my colleague Ziyong Ni and his colleagues in China invited me to join them, as I had done on the Dragon Man paper in 2021. They invited me to join in the study of this reconstruction that they've done of Jungian 2. So they got good quality CT data, basically took the thing to pieces and then virtually reconstructed it. And we end up with quite a large skull and, yes, certainly long and low, big brow ridges at the front, big face, big teeth, but certainly not looking like typical erectus. The back of the skull doesn't have as strong an angulation and a strong occipital torus at the back. Mastoid processes are quite large. The face is not as projecting from the vault as in some erectuses, and the brain size estimated at 1,140 is I think it's the biggest around that date. I don't think there's anything bigger for the human record than that at that age. So it's interesting and distinctive, apparently. So the question is, what was it? And the analyses that Ziyong has led using phylogenetics, so over 500 traits put into a phylogenetic analysis, and concluding that it's a member of this clade that contains the Harbin cranium, Dragon Man, Homo longi. It also contains some other Chinese fossils such as Dali and Jinushan. It contains mandibles that are thought to be Denisovan, Pengu and Jahe mandible. It also contains the fragmentary Denisovan material as well, and that's obviously based only on the teeth and the single finger bone. So not a lot to go on for Denisovans, but they all seem to fall together. Antisessile also seems to fall with this group, and it's distinguished from a Homo sapiens clade, a Homo neanderthalensis clade, and then deeper away a Heidelbergensis clade, and then an Asian erectus clade. So you've got these 5 main clades of what you could say large-brain humans in the Middle Pleistocene. It doesn't seem to be an erectus. It looks like it's basal to this Homo longi group, although we didn't call it Homo longi. It's quite distinct from Harbin, so we're cautious about calling it a distinct species, and also aware that Antisessile is related, so that's down there as well at the base of the clade. So that's kind of a summary, but lots of implications. Obviously, the age of origin it would suggest that the sapiens in Neanderthal clade must be about as ancient, at least 1 million years. And also it reaffirms this supposed sister group relationship of the Longi clade and Homo sapiens clades rather than with Neanderthals. So, of course, you'd expect the clade, if it contains Denisovans, genetically, you'd expect it to relate to the Neanderthals as the genetic data relates them. But it doesn't seem to. So that's another issue. Also, finally, where the common ancestor lived, it's not clear from this phylogeny where the common ancestor of these groups would have lived. It might even have been outside of Africa. But contrary to a lot of talk online, this doesn't, in my view, disprove the recent out-of-African story, the recent African origin story. That's still there, but you'd have to have a group, maybe 1 million years ago, coming into Africa and then continuing the evolution of sapiens there. And then the rest of the story is as we understand it. So that's my quick summary. Yeah.
Thank you. That was, let's say for lay people, I mean, I did read the paper extensively. For lay people, that was a lot to take in the paper. There was a lot of data, and I will praise you guys on the economy of your concision, because I know that science has length limits. So to get all that in was obviously probably a big chore.
Yeah, and it spent over a year in review, of course. It was about 1.5 years altogether under review. So we had a preprint of it more than a year and 3 months ago. Yeah.
All right, John, what do you think about all this?
Well, first, I really, really think it's great that that the preprint was available, because the way that things have gone in the review process. I mean, we're experiencing this with most of the work that I'm involved with. I think that everybody is. It's like even journals that you have, sort of have a reputation of being very fast in their decision making have become just very, very elongated. It's like, it used to be that you'd submit a paper and if the journal was not going to take it, you'd hear back really quickly. But nowadays it's no, you hear back months later and just for a first decision. So it's really, really tough. It's so important that we are getting sort of feedback from lots of different sources. And so, that it's nice that we have the preprint option. Hope that more people will use it. I think that was a great paper, to be honest. I think that the way that Homo erectus has been a problem category in paleoanthropology. It's a problem because it has this very long history. First discovered in the 1890s, and we live with a legacy of the way that people have thought about it for 130 years. And it's also problematic because the way that most people defined it, especially early in its sort of history, was based on primitive characteristics that are not present in humans and Neanderthals. And so the things that a lot of fossils share that we recognize as being erectus are actually not uniquely derived things that we would like to use for evidence of relationship. They're things that maybe are quite common among different versions of Homo, but were traditionally erectus. So these things about skull shape and skull size and, the angulation of different parts of the skull, we know an erectus skull when we're looking at it, but we know it because it's not a human skull or a Neanderthal skull. And that's why the Chinese record has been a challenge, because they're not Neanderthals, but they, but they sometimes share fossil characteristics with other fossils that have been classified as erectus. People talked about derived erectus in China for a very long time. It's really a positive thing to have identified that there's a derived genetic group. We can identify a couple of the fossils that relate to it because of ancient DNA evidence. The Harbin fossil, tremendously important in this. And now, trying to revise that record by going back and saying, when does this get started? And what derived features are there of an East Asian record that we can recognize and connect with later skulls and maybe find some lineage? I think it's really valuable. And so I think it's cool. That being said, it is really wild that we have a morphological tree that does not connect Neanderthals and Denisovans, and connects rather Denisovans closer with antecessor, and that puts quite a long chronology on the diversification of Denisovans, Neanderthals, and modern humans compared to what has been estimated from DNA. And so, I don't think that I don't expect that the morphological data are going to tell me the same thing as the genetic data. But if they tell me different things, I usually am going to believe the genetic data first and then try to work out, okay, what's going on with morphology here that is, that may explain what the discrepancy is. Because morphology is something that is genetically complex and also, is something that is going to pick up natural selection and ecological differences in a way that genetics, we can kind of sort out. And so that's where I am on the paper.
Yeah, Chris, jump in.
Yeah, so obviously, I appreciate those comments, of course, about the mismatch potentially of the morphology and the DNA. But of course, I've been asked this quite a lot already, including I gave a talk last Tuesday, so there was a lot of discussion of it. I gave a talk at a conference on Friday. There was a lot of discussion. So I've been through this a few times, and because I'm not a geneticist, so I've got a rather naive view that when we have a single thing like Denisova 25 coming along, already it changes the origin date. So now the split time of sapiens and Denisovans Neanderthals, maybe 800,000 now instead of 600,000. So already on one fossil, one bit of DNA, it's gone back 200,000 years maybe. And there's much more evidence, again, of interbreeding between the Denisovan and the Neanderthal lineages. So naively, I think that if we essentially think of, three clades really, and they're close to a trichotomy maybe, but one of those clades, the sapiens clade, goes into Africa or stays in Africa, and the other two don't. And those two then mix a lot over the next million years. Genetically, won't that make them seem to be more like sister groups? I mean, that's just a question for both of you guys. Is that, am I being naive there, or could it be part of the story?
Yeah, go, John.
I think it's totally credible, right? If you had no other information besides I'm making a measure of genetic difference, then it's absolutely correct, right? That the mixture of two lineages is going to bring them closer together. Then it will bring them closer together over time, as long as they're mixing, right? The strengths of some of the high coverage genome evidence is that you have another piece of information besides a measure of genetic distance, and that is that the lengths of haplotypes that are shared. And you can do something with that to try to talk about when mixture happened and whether mixture is recent relative to the distant common ancestor of two lineages, or whether it's happening continuously. So far on those measures, right, that's where the 800,000 year estimate comes from, is this idea that we can actually look at the lengths of things and get an idea of when gene flow last happened. But there's a big piece of complexity in it, and this is one that geneticists know about and don't communicate as well, which is that most of the genetic difference between you and a Neanderthal, between me and a Denisovan, is genetic difference that accumulated before the lineage is diversified, right? So it's what's called incomplete lineage sorting, or this, the original population that gave rise to these descendants had variation, and the different descendant lineages inherited different shares of that variation. So when we look at a gene tree, it's going to be longer than the tree relating those population lineages. And how much longer depends on how big that ancestral population was. It depends on how diversified it may have been in geographic terms. Those are things we don't know for these populations. This is where the fossil record is so essential. And so if we discover that a larger share of the human Neanderthal difference, the Denisovan Neanderthal difference, is coming from this more ancient component, the incomplete lineage sorting component, then that date could shift quite a lot, because the gene trees are going back 1.5 million, sometimes longer, and the population tree must be younger than that, but how much younger depends on those factors related to that ancestral group.
Yeah, go, Chris.
Okay, yeah, and just to pick up on a couple of points, obviously even the genetics data don't, there are different interpretations of that too, as you know, John. So, for example, the mitochondrial DNA and the Y chromosomes give you a different story to the whole genome, so that's already a difference. And there are geneticists, of course, who have quite different models to the ones we've been talking about. So we think of Ragsdale and colleagues and Cousins and colleagues, and they have really deep splits in sapiens going back more than 1 million years. So it's not as though there's a consensus even on interpreting the genetic data. And back on the morphology, if we look at, if Harbin is a Denisovan as well, and we compare it with Neanderthal, yeah, there are similarities, long, low cranial vault, big brain, big brow ridge at the front, but many differences. And morphologically, it really doesn't look very Neanderthal, particularly in the face. And so, and the finger bone too, even that single finger bone from Denisova Cave is more like sapiens than it's like a Neanderthal finger bone. So even that little bit, and the Genuthan specimen, which on our analyses is also in that group, that has the same shaped little finger bone as the Denisova Cave one, and more like sapiens. So there is stuff in the morphology that supports this, assignment. But as I say, the reality could even be something like a trichotomy, and then it gets kind of distorted by gene flow and by divergences, and also even convergences. So is that face of Harbin, the retraction under the cranial vault, is that a parallel thing that's happened independently in the Longi lineage and the Sapiens lineage? So those are all complexities to think about.
Well, so okay, I wanna jump in here then. What we're talking about here is, kind of like the fossil morphological, tree generating from characters, which these methods have been around for forever, and they were ported over to genetics in many ways. Actually, there's no difference. The characters are characters, genes are characters, morphology is characters, and what we're wrangling here is like, how do we reconcile possible different trees, and where are the trees coming from, and how complex is the internal structure. So I don't want to get into this because this is like very complicated, but Chris, you dropped like the reference to the Cousins paper and some of Aaron Ragsdale's work. Basically what that's going on is, there are alternative models for the origins of African humans that involve deep population structure and admixture. So rather than a bifurcating tree like we try to imagine, there could be more like a trellis or some sort of graph where there's deep lineages that converge. And so I think in the Cousins paper, which I think I wrote about for my Substack, but the Cousins paper showed, for example, Neanderthals are actually with Neanderthals and Denisovans, I think. I remember the Denisovans, but they're within a lineage of Africans, and then there's a deeper lineage of Africans that merges with our ancestral lineage. And so describing this verbally is extremely difficult. But I do want to throw that out there in terms of maybe it's just more complicated, and that's why our genetic and morphological trees are not because we're trying to simplify very, very complex dynamics possibly. So speaking of which, not complexification, but how things are simple. We haven't talked explicitly about this, but I think it's looming in the background. We have this new preprint out with the second high quality Denisovan genome, and so we mean high quality. It's like 24x fold coverage, so it's like medical grade. So this is Denisova 25 along with Denisova 3. Denisova 3 was discovered 65,000 years ago, or it was dated to 65,000 years ago. This one, they use both genetic and I think like some stratigraphic methods. It's kind of about 200,000. So it's at the base of when we can imagine modern humans, kind of emerging in Africa, but long before the out of Africa. And so, the great thing is we now have, I wrote a post about this, so this will be out by the time you guys are this podcast out. It's kind of like now we can go stereoscopic. We got two high quality genomes. One of the things that is in that preprint right now is they had a method which really there's this hypothesis of super archaic admixture. This would be from Homo erectus or whatever. Again, like the fossil record in East Asia is still labeling things, right? But archaic, pre-Denisovan, pre-Neanderthovan populations, and this result has been, I think it goes back to 2014 with Sirian Sakaram's paper, actually, out of the Reich lab. But I think this was like when he was starting at UCLA. In any case, the other hypothesis, Neanderthals have modern African human admixture. And so that's why there's a statistical artifact of looking like there's older stuff in Denisovans, right? Anyway, in the preprint, they compare Denisovan 25 and Denisovan 3, and that super archaic stuff looked like the ones, the alleles that they identified as super archaic. So these are alleles where they are the outgroup to Neanderthals and Africans. Okay, those alleles show variation in linkage disequilibrium over all of the 5,000 generations, which basically means that's evidence of some sort of pulse admixture event before 200,000 years ago. So this is not the final word, but this is, I think, leaning toward the hypothesis that Denisovans had admixture from other human lineages, probably the relatives of, say, Demonisi, Homo georgicus, all these other Homo groups that were there many, many million, well over 1 million years earlier, right? I think that is also something that is important to talk about when we're talking about these morphological characteristics, because who knows where these morphological characteristics came from, right? So I will just actually, John, why don't you start, and Chris, we can go to you.
Yeah, I think the cool thing about this genome is it is such a good reminder that, the thing is that it doesn't look like the lineal ancestor of the later Denisova 3, right? It looks like it is a descendant of a population branch that has a much earlier connection with the Denisova 3 branch. And we already knew this about Denisovans. We knew that they were a diverse group. We knew this because the genetics that today's people have inherited from them, especially people in island Southeast Asia, Papua New Guinea, the Philippines, is variation that appears to have a deeper connection with the high quality Denisova 3 genome than if they had descended directly from that Siberian population. So we knew that there was population differentiation among this group that has been identified genetically. But the thing that it reminds us is that we are only sampling DNA from the very extreme northern part of the hominin range. And the mainstream of these populations was almost certainly in areas that we aren't genetically able to sample, places further south. South Asia, Southeast Asia, Southern China, maybe Southwest Asia, right? These are places that should have had larger hominin populations. They should have been more compatible with hominin life, and where we should have greater potential of morphological, ecological variation. But we also haven't sampled those places very well with fossils. So both the fossils and the genetics are telling us about a part of the hominin range that may have been quite peripheral in a lot of the time periods that we're interested in. We're trying to piece together the story of how these things are connected, knowing that there must be missing populations, right? Populations that are not represented in our current fossil array, and knowing that the genetic mainstream, right, that the extent to which there was differentiation genetically was happening because of common ancestors in these places that we haven't sampled.
Yeah, Chris?
Yeah, I agree with that, John. Obviously, yeah, even for looking at Harbin, I mean, that is right up in northeast China. It's one of the coldest places in the winter in China. Average temperature is -16. Now, that is extremely cold in the winter, and that's now. They may have been even colder in a glacial stage. Those people were adapted to extreme cold much more than Neanderthals seem to have been. And we get the same thing in Jinushan. That woman's pelvis is really broad, short and wide. I think she's, Karen Rosenberg and Wu said that it's the biggest pelvis reconstructed in the human fossil record, wide in terms of width. So that group are cold adapted. The ones in Denisova Cave are probably similar. The ones at Jahe up on the Tibetan Plateau are probably similar. But as John says, down south, in India maybe, in Southeast Asia, in island Southeast Asia, they're going to be different. And they're going to be probably very different, with hundreds of thousands of years of divergence from the ones we know about. So we've got to bear that in mind. It may be that the Denisovans were much more varied than the Neanderthals were, and also even more varied in their adaptations. They're up in cold steppe tundra in the north, and they're down in tropical, forests even potentially down in the south.
Yeah, I want to reiterate this because I think, we're talking about, in this podcast, we are talking about things that are, somewhat speculative on the frontier, obviously, in some ways, we're for example, like, really, really interrogating the phylogenetic tree and the relationships. But there are other things that I think we're converging on kind of a consensus. So, for example, with the Denisovans, which you guys are talking about, is you look at the Denisovan whole genome, the Siberian Denisovans. Now we got two of them, two whole genomes. And so when you have two whole genomes at high quality, you have really, really, really good data on these sequences. And what they have done in the past, decade or so, with Denisova 3, and now they can do this with Denisova 25, is they can look at the genome and compare those Denisovan segments to inferred Denisovan segments in various human populations. So Papuans are about 4% Denisovans. The Aita, Negritos in the Philippines are about 5% Denisovans. There's like 0.2% Denisovan in South Asians, 0.15% in East Asians, and so forth. Compare the segments, and what they find, and again, you have 3 billion base pairs, and you have millions and millions of markers across those base pairs. You have a lot of diversity. So, even 0.25% you can analyze. That's still a lot of markers. What they found is when you compare the genetic distance between the reference genomes, which we have the ancient DNA, we're not inferring anything, and you compare them to the inferred Denisovans, that's why Chris is saying, oh, these like populations are split hundreds of thousands of years ago, because that's the mutational distance across the segments, right? And so what we're doing is we have these Siberian references, and we can compare them by distance. And, some of these distances are incredibly, conservatively 200,000. I've seen estimates as big as like almost 400,000, which means there was diversification possibly of the Denisovan hominins at the root, very close to the root of their separation from Neanderthals, So they went into East Asia. And I think what we want to emphasize here is like, just look at the geography, that hasn't changed that much. You go from Mongolia to, what was then Sundaland, Southeast Asia. The climatic ecological shifts there are very, very radically, just incredible compared to, Neanderthals, which we now know that there are deep Neanderthal lineages. And this preprint actually also infers there was at least a third deep Neanderthal lineage, but we can talk about that maybe later. But Neanderthals occupied this sort of kind of, high latitude, biome. Whereas Denisovans occupied all of these ranges. So of course they're going to probably be more diverse, probably physically, local adaptations. In terms of super archaic admixture, we know that there's like hominins in Southeast Asia that they're probably different from the ones in Northeast Asia and Siberia. So that would be another local factor. And I think the final thing that I have mentioned before is in my reading, one of the things is because of the way, East Asia, mainland East Asia, China, Southeast Asia, they're structured in the way the floral diversity is actually like much higher than in Europe, because in Europe, you can't retreat. The Mediterranean's there. I think that this is probably something that allowed the Denisovans to stay more diverse, at least morphologically in many ways, because they could retreat. The warm weather Denisovans could retreat, whereas with Neanderthals, they couldn't go south of Israel because there were other people there, and that were blocking them off. So I think these two differences between Denisovans and Neanderthals are going to require further exploration. And so, maybe they are sister lineages, maybe they're not, but whether they're not, their geographical context, their ecological, climatological context is very different. And I think that's important to note.
I think also for Neanderthals, we really don't know how far they ranged to the east. Did they ever get to China? Who was in India for all of this time? We just got that one fossil, really significant fossil from Namada. Our analyses didn't place it anywhere. It kind of sat on its own, and so did Xinjiang. So I'd have liked it to have fallen somewhere nice like Heidelbergensis or even with Longi, but it didn't. So who was in India? And that's the huge refugium too. And we really have very little idea who was there.
Well, just again, going back to geography, how you go east, there's possibly a northern route, and the preprint actually alludes to the possibility of a northern route for East Asians, but definitely there's a southern route that goes through India, and our lack of archaeological, a paleoanthropological, visibility there is really a tragedy.
It's funny because the southern tier of Asia, India, Pakistan, Iran, Arabia even, play a really big role in the way that a lot of us think about the dispersal of humans, right? At different levels, right? The out of Africa 1, the out of Africa 2, right? Out of Africa, 1.5 probably is what's going on with these Denisovan Neanderthal ancestors. We know that there were large-scale dispersals, and this area plays a large role in our thinking about it. And it has often been the case that we think about populations having been there and acting as a block in some ways to prevent dispersal across parts of that region. The reason people talk about the northern route is the idea that there's something standing in the way of the southern route. And yet we really don't know who was there. And that is such a big challenge for us.
Yeah.
And such a big challenge to the archaeological field to, sort of build up better evidence about the populations of that region.
And also the paleoclimatic signal, we know that some of those areas were very arid at times. You've got the Thar Desert to the west of India. You've got huge areas of Arabia that at times were green, at other times were completely arid. So there'd have been pulses of opportunity of times to people to cross and other times when they couldn't cross. So yeah, that also is a big factor which we need to learn more about.
Well, I mean, I think, obviously there's been some massive changes over the last couple of million years, over the last 100,000 years, et cetera, during the Holocene. But I do want to reiterate again, going back to geography, Asia has this big, mountainous, arid heart. And, today, if you look at the genetics, there's an east and there's a west. Like South Asia is a little bit of a bridge, but, outside the purview of this podcast, but its modern admixture patterns are actually relatively late. It looks like for most of modern human history, South Asia, east of say like the Indus River or like the Iranian Plateau was more part of Eastern Eurasian genetic continuity. And with Denisovans versus Neanderthals, I think most people assume, as you said, like we have like lacunae here, we don't necessarily know. I think most people assume, though, because of the admixture patterns, some of which are in the preprint, and I want to get back to that, that a Denisovan related or Denisovan population was in South Asia and Neanderthals were in Iran. So this division between South Asia and its association with Eastern Eurasia seems to be pretty old, and I'm assuming actually, for most of the last million years or so, or more than a million years, 2 million years, been ice age. It's been very dry, very cold. I think the Thar Desert does get bigger. So it was actually a bigger ecological barrier in the past than it is today.
Yeah, and there's that interesting paper by Ruan and Timmerman and colleagues a while back, I think earlier this year maybe, where they modelled the potential ranges of Denisovans and Neanderthals based on where the sites were. And they had several different ways of doing it, but a lot of the data suggested Denisovans potentially could have ranged a long way across Asia, even towards Europe. They could have even made Eastern Europe. Now, obviously, that's theoretical modelling, but we should bear in mind that there are sites right across Russia with caves, with stone tools, and we don't have any fossils, but sediment DNA maybe can start to map who was there. And of course, we haven't mentioned Heidelbergensis, and on our analysis, Heidelbergensis is also around for a lot of this time, down to about 300,000, who knows how much later. So that's also in the picture. Did that ever get over towards China, or was that just a Western thing? So there's even that complexity. But I would say Denisovans, the range we've got, even extending it southwards and upwards to Harbin and Denisovan, that's the minimum range. At times, they must have expanded further than that.
Sure. Well, I mean, so I want to talk about the admixture of Denisovans into modern humans and what that tells us about modern human admixture or expansion. Okay, so the preprint makes some claims, which I think that they're disputable, but I mean, hey, they're based on real data and real results. And so basically, it kind of confirms a repeated pattern. So we have a new genome. So having the second genome, by the way, it looks like it really allowed them to capture more Denisovan variation, because obviously a single genome is only going to capture a subset of the variation within a population. So you have a separate genome that is 35,000 whatever years older. You're going to have like more variation. They captured more Denisovan segments. They picked up low levels in European populations, by the way. Probably, I mean, they believe that it's mostly gene flow from East Asia. Because the ancient DNA, the ancient Europeans don't show it except for one sample. But they have at least 2 admixture events in East Asia. This is kind of agreeing with work that goes back, Browning et al., goes back almost a decade now. Okay, so it looks like one of them is very similar to Denisovan 3, the classical canonical Denisovan, and the other one is related, but somewhat distant. So more like, the Denisovan 25, Denisovan 3 clade together. Okay, and then they also found in South Asians and Oceanians admixture from what I call I'll call basal Denisovans. I know that's again, it's privileging our northern samples, but hey, we got to go with where we got to go. And so these Denisovans diverge 200,000 or more. These are the ones that are, that's why I call it basal, deeply diverged. So these are southern Denisovans, right? And they estimated, again, having more data allowed them to do some estimations that are more robust of things like the admixture dates, again, looking at the linkage between Denisovan alleles. And they estimated that the admixture of Denisovans into Oceanians was 10% more recent than the admixture of Neanderthal ancestry into Denisovans. So this is important because I think we have a pretty good estimate of Neanderthal admixture now, right? It's about 48, it's between 45 and 50,000, right? I mean, what do you guys say? It's around there, right? Probably not much earlier than 50, definitely not later than 45. It's within that interval. So if it's 10%, well, then it's 40 to 45,000 years ago. Okay, so we have that now. But the admixture for the other two, for South Asians and East Asians is 20 and 30% later than Neanderthals. So we know that Neanderthals is a relatively simple story. Admixture probably in the Iranian plateau or the Levant of some out of Africa group. And so that is our date peg there. And so now we have multiple different Denisovan admixtures. And this is not a novel result from this preprint. This has been a repeated finding that there's multiple different admixtures. And so we have this complexity in East and South Eurasia, versus a much more simple narrative about West Eurasia with Neanderthals. And I know that there are separate admixture events with Oasi in Romania, but it doesn't look like that they left a long-term impact. When it comes to Neanderthal admixture all over the world, it looks like it was one Neanderthal population from the Middle East that contributed the overwhelming amount of the genetic content, right? Whereas with Denisovans, South Asians have South Asian Denisovan. It looks like there's another paper that implies that. And in this paper, they looked at the admixture into South Asians and Oceanians. One, they occurred at different times, okay? And secondarily, even though it looks like it's from this basal Eurasian population or basal Denisovan population, the recombination pattern, the breakpoints of the segments are very different, which implies, again, a separate admixture event. And so, this is probably simpler than the reality, but what you're seeing is okay, same population, different admixture times, different admixture events. In Northeast Asia, you have another later admixture event, probably the one related to Denisovan 3. And then, Chris, you were talking about how Denisovans, may have ranged further west than we thought. This paper, and there are other papers that imply this, that there's a baseline common Denisovan admixture across Asia. This paper kind of implies that. Oh, I mean, if that's true, then I think the implication there would be it's somewhere further west where the IUP, the initial Upper Paleolithic, were expanding eastward. Because if it's in all Asians, but it's actually not, and I don't know if we're going to get to this, but there's a new paper about the Jomon, and it shows that the Jomon might have had contributions from an East Asian population that didn't get Denisovan admixture, which again, we have all these simple models that are elegant, but it's quite, to me, if there's an East Asian population that doesn't have Denisovan admixture, we're talking about a very complex, historically contingent set of admixture events that can't fit onto a 3-twig tree, like we would do. You know what I'm gonna say. Anyway, I threw a lot at you, John. You're laughing, so I want to let you speak, and then Chris, you can talk.
I think I'm laughing because if we had these sorts of results for Neanderthals in 2005. We would have understood them immediately, right? It would have been, well, okay, the modern humans show up in Europe, they mix with Neanderthals. There's a series of mixing events as they sort of inhabit the whole continent. And right, that's sort of the way that we thought that things would turn out. And in fact, it was always puzzling that once you found that there was Neanderthal admixture, right after 2010, when you were like, okay, well, there's a low level of Neanderthal mixture into everybody's ancestry, it was puzzling that it was the same. That didn't make any sense. We had geneticists trying to understand how it could be that a population could disperse through the range that Neanderthals were in and not pick up any more Neanderthal, right? So we knew that was a problem. It's still sort of a problem, But it's a problem that's probably partway solved by the dynamics of the earliest immigrants into Europe and Northern Central Asia who didn't survive or didn't persist in sort of a substantial proportion in later populations. So
Yeah.
Although the region was not literally depopulated, it was populated by people that didn't play a huge genetic role in later groups. And so we understand a bit now how that happened. We can see from the Denisovan evidence that didn't happen that way in East Asia, Southeast Asia. It instead happened the way that we sort of expect that a dispersing population into the range of existing populations might look. Now, in fact, maybe it still remains a bit of a puzzle, right? Why people don't end up with more. We sort of expect a wave front of modern humans moving into a range where they can interbreed with the existing populations. Let's say they're Denisovans. If they're moving into this, they ought to pick up more as they go. They don't pick up much, right? They pick up a little bit in what appear to be spurts. That may be, again, in part due to the fact that the earliest people that existed there, the earliest modern humans, probably had a high extinction rate or low transmission rate of their ancestry to later groups in the same way that they did in Europe. So we're not today sampling the earliest initial Upper Paleolithic people very well in today's genes. Right? We have a lot of events have happened in between, and a lot of those early people were not super successful. But some of them were massively successful. And the particular times and places that they mix with Denisovans are what we're picking up today. It is great, I think, that we're able to look back at these populations from multiple viewpoints today. Right? If you took an astronomical analogy, this is like we're looking at very ancient events, and there's a massive galaxy in the way. And it blocks our view, but we get, because of the Einstein lens effect, little glimpses of what's going on behind that. This is kind of what's going on with today's genetics. The massive events that happened are the end of the last glaciation and the beginnings of agriculture. They're blocking a lot of our ability to see initial Upper Paleolithic stuff. But we are getting multiple views of that stuff because multiple groups survived.
Yeah, Chris, jump in.
Yeah, so back to the Yomon, which I'd really like your impressions, you two, on where could these people have been where they didn't get Denisovan integration? Where were they? Do you have any guesses?
I wrote a post. I mean, it'll come out. Like I think they probably had to use some northern route. Because I've tried to read the Japanese. There's some Japanese ethnogenesis stuff. Let's set aside like the more recent ancestry, which we know is, I'm going to say it, Korean. But let's focus on the Yomon. That ancestry, they often label as Paleo-Siberian and Basal East Eurasian or something like that. It's not really well modeled. It seems to have some affinity to Southeast Asian populations, but then it also has affinity to Siberian, deeply diverse Siberian populations. With their Denisovan admixture fraction, assuming that this is correct, that the estimate is correct, they almost certainly have to be a merger of some sort of more conventional East Asian population, because they have the two admixture events that you see in mainland East Asia. Okay, so that's confirmed. But their fraction is lower. Okay, the easiest way to explain that is a basal Eurasian type of explanation, right? So basal East Eurasian. Where would they be? My hypothesis, I don't think they're going to be in Southeast Asia, because it looks like Denisovans are crawling all over the place there. There's people there. I think it's got to be, it has to be like a northern route through Siberia. You guys are the, paleoanthropologists, but my understanding is, our own lineage of modern humans out of Africa were culturally more flexible, and we pushed north into the tundra further than our cousins did. And so I would not be surprised if some modern humans of the initial Upper Paleolithic come up through Central Asia and sweep up through Mongolia, through areas. I memean, obviously Denisova Cave is in liminal Mongolia, but further north. And also, as John was saying, there's all these different interactions. Just because a population is in the same area as another population also doesn't mean that it needs to mix. They could have just gone through. right? And so what I'm looking at is I think it's probably a northern route just because the population density is going to be lower. The human occupation is going to be patchier. And we do know that the Jomon have like some sort of connection to Siberia. It's just too clear. It's just they don't fit into the standard models, right? And I think that's partly because there are basal East Eurasian groups that went extinct. I think this is probably, to be honest, connected in some way to the Australo-Melanesian signal that you see in South America. These are not Australian like sailors, right? Like there's something going on in the heart of Eurasia where we lost population structure and admixture. And, this is kind of alluding to what John said earlier. Like we're seeing, through the mirror darkly, just through the very, very recent past in our current structure, right? Anyway, John, what do you think?
I think it's a problem. I really, the thing that I think about with this is that there's this really cool recent work on what's called the North American Upper Paleolithic or the American Upper Paleolithic, right? These early North American artifact types, like at Cooper's Ferry in Idaho, that are connected with ultimately early artifacts in Japan. And you have this sort of commonality of that, sort of coastal Northeast Asian and coastal Northwest US populations at the time of the earliest inhabitation of North America. So I'm looking at that and thinking, I'm looking for the source populations of the Americans ultimately, and they're in that area of Japan, Mongolia. We know that there's genetic connections of those people. Right? And the problem with the Jomon, now these are not Jomon people, right? Jomon people are a later arrival into Japan. But the problem is that if I've got sort of the ancestors of Native American peoples in this part of Northeast Asia at the last glacial maximum and thereabouts, and they've got the typical amount of Denisovan, where are people that wouldn't have that, right? Are they further north? I think an extreme northern route could in fact be possible. But if so, where are they? How do they get to where they are now? How do they create the mix that is the Jomon mix? It's, it's a problem.
I mean, I want to say, this is an interesting issue because, Japan is a country that has like a relatively advanced genetics infrastructure. They have relatively advanced like public medical health infrastructure. So we have a lot of data. We have a lot of we have a lot of work to try to figure out the origins of like the Paleo Japanese. I'll use that word, right? And it's just not totally clear. Like in Europe, like, in Ireland, like another island, or Britain, we kind of know, bell beakers, and before that, the Neolithic. We got it. It's taken care of. Japan, it's actually not as clear, partly because we just don't know what was going on in mainland Asia. One of the things in the paper, in the Cell paper that I'm alluding to here, but in general, it's like there's an affinity to coastal East Asians among the Japanese. Well, of course, they're on the coast, but like, what does this even mean? The Korean part of their ancestry is really straightforward. It's the Jomon ancestry. There's stuff in there that basically our models cannot explain. And that is why they're throwing up these because like the different papers, you see them publishing contradictory things. And when that happens, traditionally, it's because their models are missing parameters or variables that would just like snap it into focus. And so we're missing something, and we're kind of like groping in the dark to try to answer that question. And now there's this new twist where they might have less Denisovan ancestry, which means, the history of East Asia, wow, it was like really complicated. It was like more complicated, partly because there weren't like as many replacements. I think that's one of the answers here. Again, I think the same dynamic that's operative to Denisovans is operative to modern humans, where East Asia has this like range in geographic depth, where the older population, more like anciently resident populations aren't totally replaced by newcomers. Whereas in Europe, they were until like basically the last wave. Chris, jump in.
Yeah. So switching it back to Ireland, Southeast Asia as well, I mean, there are contradictory signals I'm picking up about whether the Denisovans were down there and to what extent, because some of the authors of the Denisova 25 paper seem to be implying, well, those diverse groups could have been in India or in Southern Asia, but not necessarily a long way to the south. But then you've got people like Jacobs and colleagues who are saying, well, actually, we think there's introgression right next to Papua New Guinea as late as 30,000 years ago. Now, what do you two guys think about that? Those different views?
John, you haven't talked in a while.
Yeah.
Well, so
Over to
I
you.
think that from my point of view, right, the problem that all of us have to grapple with is that all the Neanderthal ancestry happened around 45 to 50,000 years ago, right? And so everybody everybody outside of Africa who has Neanderthal ancestry has got this shared event in their lineage,
That's
and
right.
it is the overwhelming majority of their genetics. And that means that all the people in PNG and all the people in Australia and all the people across the Pacific and all of them came from this ancestral group that was 45 to 50,000 years old. And we have thought that there's archaeological evidence of arrival of modern humans before that in a lot of these areas. And so that's a mismatch, right? The evidence from Laos, it's a mismatch. The evidence from, the Majabebe, early Australian, mismatch. And so either the mismatches have to work out as, well, I guess we were wrong about the dates of those things, or they have to work out as this is somebody else. Maybe an early modern lineage that is not the major contributor to today's groups, or maybe somebody archaic that we just have misclassified, or maybe some third option.
Well,
But it's a mismatch.
yeah,
And
yeah.
we actually haven't worked through that mismatch.
Yeah, I will say I definitely lean towards the idea that Southern Asia was more accessible to early African humans, and that those modern sites, we use the word modern, but STEM human, whatever you want to say, that led to us. The Altai Neanderthal sample, which I think dates to 120,000 years ago, that genome, and is that Denisova 5? Anyway, it's one of the Denisovans labeled, but it's not a Denisovan. It's a Neanderthal. That Neanderthal, like a lot of Neanderthals, has modern human gene flow. And it's from a basal root population that left early, obviously. I mean, it didn't the Denisovans were not, the Neanderthals were not in Africa. So that means that someone was out there.
This is why I'm answering Chris's question in that way, right? Because if there are African-derived modern-related populations in Laos and across Southeast Asia and as far as Indonesia, when the people with the Neanderthal genes show up, then the Denisovans have got to be very remote.
Yeah.
In that scenario, right? They've got to be somewhere quite distant. The alternative point of view is that actually Sahul, New Guinea, Australia, was inhabited before 50,000 years ago by Denisovans. So these are all open possibilities for us right now. I don't think that we have the evidence to say one way or another, but you can see that the more that there is pre-priming of an African related set of populations into Southeast Asia, the more Denisovans have got to be distant or peripheral when the Neanderthal descendant modern humans show up.
Yeah. So just real quickly, in the preprint, in the Denisovan preprint, 2025 preprint, they say that the very very short, so it's only 10% difference Neanderthal admixture to Denisovan admixture. That this is evidence of a southern wave that's separate from the primary wave. I don't necessarily think that that's true, just because, first of all, we know that, as John said, the Neanderthal admixture happened at one time. They assume that in there too, right? So they're from the same initial Upper Paleolithic. The second thing is it could be, and some people have said this, and there are many, not many, but there's a handful of papers now. It's a consistent result that the Oceanians have a late admixture. They have complex, let's just say complex Denisovan admixture. There's something going on there. It could be that it's I think John has said this too, but I mean, a very, very Denisovan enriched modern population. So it could be 70% modern, 30% Denisovan. They are not part of the initial Upper Paleolithic. They're there in Southeast Asia, and they get absorbed. And, the way that these statistical inferences work, I mean, they only have a certain amount of power to detect things. So, for example, and John remembers this. I mean, you remember this too, Chris. It was the genetics was not powerful enough to detect the Neanderthal admixture before we had a Neanderthal genome. And then all of a sudden we had a Neanderthal genome. We had so much data that like our precision went way up. And all of a sudden they're like, oh, but it's because it was only 2%, which like was not detectable. But if you read the papers before that period, they'll be like, oh, well, we've rejected this hypothesis because, well, it's because they didn't have the statistical power. So maybe there's some modern pre-IUP, modern human ancestry in Papuans. Maybe it's like 10 to 15%, but maybe it's hard to detect because, first of all, they're like much closer, to the IUP anyway. And so, we'll get an ancient genome and it'll make it all clear. I mean, that's what I'm hoping. What do you guys think?
Yeah, I mean, ancient genomes from down there would be wonderful. And I think places like Papua New Guinea, because there are real highland areas there where it's much cooler. And maybe there are some caves up there that really would have some DNA surviving. So places like that could be promising. And I think, Tampaling and obviously Lida Ager, those are modern teeth at Lida Ager. I wish they were directly dated. But anyway, there is a hint that there was a sapiens group there before 70,000, and then you've, as you say, Madjabebi too. If that's accurate, who the hell was there at 65,000? And of course, over in the West, we've got the Epidima Cave, and I was involved with studying that. And there, apparently, it's only the back of a skull, but that Epidima fossil looks like a Homo sapiens just from the back of the skull, or at least not a Neanderthal anyway.
This is in Greece, right? This is in Greece,
yeah. And it's over 200,000 years old.
Yeah.
And of course, if the signal of Neanderthal interbreeding is also correct, that has this changeover of mitochondrial DNA and Y chromosome, maybe 300,000, Epidima could be part of that. And also, if they got to Greece, maybe they did go eastwards as well. If the conditions were right. So there could even be a very early sapiens thing, which mostly or almost completely disappeared from the current data.
Yeah, and I want to say, like you alluded to the mitochondrial and the mtDNA. And so the issue here is the mitochondrial and mtDNA of Neanderthals are quite distinct from modern humans. They're not part of the modern human like haplogroup lineage, but they're also quite distinct from Denisovans. And, the best working model right now is that they come from admixture or introgression from I'll just say Paleo-African lineage, part of our own lineage, but earlier, like at the base, maybe like 200, 300,000 years ago, right? Obviously well before the out of Africa, but perhaps even before the dynamics that led to the emergence of anatomically modern humans, could before all that stuff, right? And that's there. It's kind of weird that the autosomal DNA is not replaced and the mitochondrial and the Y is. And, David Reich has said on multiple occasions now, maybe we just don't know what's going on. Maybe we're just all wrong.
I know he doesn't believe that replacement at 300,000 of the mitochondrial DNA and the Y. He said to me the chances of that happening are absolutely minute. If there was gene flow at 5% level, why would you get complete replacement of mitochondrial Y? Selection, maybe,
Yeah, it could be selected because why is it that the Y and the mtD, the two uniparentals? Because so it's a 5%, so the way it works in population genetics, a 5% chance at time t, there's a 5% chance of fixation, right? But if it's 5% and 5%, you multiply that as independent probability. What's going on there? I think that's what David's the math doesn't work out, right, John?
Well, I know that's true. This has got to be my last word, I'm afraid, because I've got to go and teach a class. This has been amazing. This has been great, and we should do it again. I know David reads my work, but he probably doesn't read the work that I published in edited volumes in 2005, where I describe how this could happen to mitochondrial DNA. Right? It's not a big mystery that some genes are affected by selection, and if you have, especially a uniparental system, where the effective size is low because it's inherited from one parent and not both. And so you have a lower sort of pool of variation to begin with, but you also have both of those systems are few genes that matter, and the ones that matter are easy to break. And so in a smaller population with lower effective size over time, they tend more toward a mutational ratchet effect of sort of things, slightly deleterious things build up. That means that anytime you have contact from another larger group that has a larger pool of variation, somebody's haplotype that doesn't have slightly deleterious variants has a huge advantage. And so I think that those systems are not hard to explain. What is interesting that we see again and again is these these contexts between populations lead to 2%, 3%, maximum 5 or 6% gene flow. And that's interesting that there's a consistency when we have seen evidence of these population contexts. Now, it may just be because the few cases in more recent history where you have 25, 30%, at a large enough distance, those blend together and you don't notice that, oh, we had this small fraction from somebody, right? There's an effect of maybe we haven't quite mastered the measurement of ancient gene flow in a way that would let us talk about these dynamics, the evidence that we have about them. But it is interesting, I think, looking back, that the dynamics of these populations were long separations with little gene flow, occasional expansions, dispersals with small fractions of gene flow when it happens. And yet there is, coming back to the Yunshan case, and yet there are these morphological things that lead us to think that populations are connected. And that's an interesting pattern. It's interesting that the morphology might actually be a sensitive sort of measurement of this in some way. It's interesting that the fossils have the potential of providing information that is different from the genetics. And it's interesting that both of us on both sides, genetic side, the fossil side, are just waiting for the next discovery. The next discovery is going to change everything.
Yeah, it's a great position to be. All right, I'll let you go, John. Chris, do you wanna close out?
Well, I've enjoyed this discussion, and there's so much more we didn't get to, so we can always try and do it again. Yeah.
I mean, I'm sure there'll be more papers, more findings, and I don't even. I'm gonna end it here because we just go on forever. But it was great talking to you guys and catching up. And you guys have a good day.
Great, thank you.
Thanks, Razib. Thanks, Chris.
This podcast for kids